Hello everyone, my name is Faiza and I'm working on a PhD thesis on the quinone reductase 2 enzyme or NQ2. Quinone reductase enzymes are an ancient class of flavoenzymes which catalyze the detoxification 2-electron reduction of quinones to a dihydroquinone as a way to bypass the formation of semiquinone, which can produce ROS. Humans have two quinone reductase enzymes, NQ1 and NQ2. NQ1 is a classical phase 2 enzyme in that it is induced in response to oxidative stress and it metabolizes a quick and efficient quinone reduction reaction. On the other hand, NQ2 function is less clear as it does not use canonical reducing cofactors NADH or NADPH efficiently. Here is an image of NADH, a canonical cofactor for many reductases. Unlike any other reductases, NQ2-dependent catalysis relies on non-conventional, simpler nicotinamide cofactors such as NRH, and the extent to which these simpler reducing cofactors is present in cells is not clear. To illustrate the difference between NQ1 and NQ2 catalysis with canonical reducing cofactors, we did transient state studies of NQ2 with NADH. For NQ1 with other groups, they found that the reduction was a simple second-order process in that the rate of the reaction increased linearly with substrate concentration. Meanwhile, with NQ2, we found that the rate goes up until it reaches the saturation. As we could fit this data to a rapid equilibrium model, we predicted that NQ2 was binding to NADH non-productively, and of course the rate is a lot lower for NQ2. Then we asked the question, why would an enzyme evolve to be catalytically inefficient? We think that NQ2 evolved to signal instead of being an enzyme. The consequences of NQ1 loss is very different from that of NQ2. In mice, when mice are treated with menandine, which is a quinone substrate for both NQ1 and NQ2, when NQ1 was knocked out and the mice were treated with menandine, these mice had increased sensitivity to the toxic effects of menandine, which is consistent with NQ1's role in quinone detoxification. When NQ2 is knocked out and mice are treated with menandine, these mice were less sensitive to the effects of menandine. There is also more literature that shows that NQ2 inhibition led to decreased sensitivity to paraquat and that NQ2 may be involved in learning in the brain. These observations suggested that NQ2 may not just be an enzyme, but it may be doing something more like signaling. We are investigating the molecular mechanism behind NQ2 cofactor specificity by an evolutionary analysis. We found that the cofactor specificity of NQ2 in reptiles and in birds is the same as that in mammals. We think that there is functional NQ2 in all amniotes, and some amphibians and lopin fish have NQ2 but may not actually be functional. We've also discovered that NQ2 emerged around the time when vertebrates were emerging from the ocean to the land. In the future, we're going to make ancient NQ2 enzymes in the nodes that may have cofactor specificity closer to NQ1 and find residues in NQ2 that's responsible for its cofactor specificity. We're also going to look for NQ2 interacting proteins by an NQ2 biotin ligase construct and do some more proteomics to see if NQ2 may be involved in things like, say, apoptosis. Thank you.